The next session is the Pecha Kucha presentations, and just so everybody is relieved, this is a very informal session, and this is about having people present their work so you can all become familiar with what each other is doing. So our volunteers for this are going to get 5 minutes, and they're going to have 15 slides, and each slide is going to go by in 20 seconds. And there'll be no questions in between because we're encouraging you to sit down and talk with them at dinner about their work. So our first presenter is Dr. Shiva Prasad Dhar, who's coming from India, and he's going to talk about alternative clinical trial designs for research on hypertensive disorders of pregnancy in low-resource settings.
Yeah, thank you very much, and good evening to all of you. And what I'm presenting today are the issues related to 2 clinical trials that our group is involved in. The first one is a trial that we just completed, the community level intervention for preeclampsia. It's a cluster randomized controlled trial. The second trial, which is ongoing, in fact, will close out by the end of this month. It's a step-wedged design of introduction of a new blood pressure monitoring device. The community level intervention for preeclampsia was actually a collaboration with University of British Columbia and King's College London now. The objective is to reduce preeclampsia-related maternal and perinatal mortality and major morbidity by 20%, as well as adverse maternal and perinatal events using a cost-effective intervention appropriate for LMIC setting. Those are the objectives of the trial. The intervention is actually a package, which is identification of hypertensive disorders in a pregnant woman, starting at the 12 weeks of gestation and monitoring her blood pressure throughout pregnancy till delivery and postpartum period. A mobile app guided package helps the community health workers who are trained for this blood pressure monitoring using the blood pressure device in identifying the high-risk women, then bringing them to the referral facility and getting them the loading dose of Maxulf and methyl dopa, and then initiating a referral. The trial as such was done in 4 countries: India, Pakistan, Nigeria, and Mozambique. We just finished the primary analysis, and we should be publishing it pretty soon. The second, the stepwise design randomized control trial is to evaluate a new blood pressure monitoring device on a composite outcome of maternal mortality and morbidity by more than 25% in 9 LMIC countries. We had a pilot phase and then the definitive trial phase. As I said, we are just about completing the data collection for the definitive trial phase. That's the device. It has a traffic light system which alerts the health worker for both hypertension as well as shock. We trained the community health workers for recording the blood pressure. This was the first time that a clinical task was being assigned to these community health workers. Those are the trial sites. Besides India, there are a number of sites in Africa as well as one in Central America, that's in Haiti. And each site has, some of the sites have more than one hospital, so basically the unit of randomization is a hospital, so each hospital gets randomized at 3-month intervals to the intervention. And of course, till it gets randomized to the intervention, the data is collected, serves as the baseline for those clusters. The issues, especially in stepwise RCTs, are with respect to, we are collecting aggregate outcome data, so we did request for a waiver of individual consent because we are not collecting patient-level data. The contamination is avoided, and one of the major challenges is if one of those intervention clusters or one of those units drops out in the middle of the study, your power gets compromised. I'm sorry, the last two slides were busy. I couldn't do it.
Thank you so much. And then you'll have a lot to talk about the ethics that you faced at dinner with people. Thank you. So our next presenter is Stephen Balan, and he's going to talk about a quantitative framework for balancing ethical trade-offs in a vaccine study design during a highly fatal emerging infectious disease epidemics. Thanks.
Thank you, and thanks to the organizers. This has been a really fun meeting. So I'm going to be talking about a quantitative framework for balancing some ethical trade-offs in vaccine trial design. And a lot of this is on a poster that I also have back there, so feel free to take a look at that as well. And the framework that we take to look at this is similar to that in the previous session, which is to use a lot of simulation. And my focus is on how the dynamical processes underlying transmission interact with study design in ways that can help you develop better study designs, more ethical study designs, more efficient study designs. So this is showing the number of cases at the district level in the 3 affected countries of Ebola. And you can see how varied the epidemics are. And this was really challenging for designing trials because the discussions for vaccine trials started at the during the peak of the epidemic around October and November, but the trials actually were only executed towards the end of the epidemic. And the CDC in particular wanted to do a stepped wedge design so that they could both roll out the vaccine and evaluate it at the same time. And that idea was proposed at the peak of the epidemic, but the actual trial was started later. And they didn't do a stepped wedge, but the motivation was really that you wanted to balance these participant benefits to give them the vaccine and give everybody the vaccine with the societal benefits to actually evaluate a vaccine rigorously and have information that can save lives later on. And what we showed through simulation is that actually if you compare a stepped wedge to a randomized controlled trial, individual randomized controlled trial, the stepped wedge has far lower statistical power because the different areas of the country have such different epidemics, both in intensity and in trajectory, and that the power would decline so much that it would basically be futile. But stepping back, are we committing therapeutic misconception in the first place by saying we should be rolling out an intervention that hasn't even been studied? And we argue no. For very severe diseases, participants at high risk of infection are better off being vaccinated than not, even if we don't know if the vaccine works, because the extent of the side effects to counterbalance that are too unrealistic after a vaccine has emerged from a phase 2 trial. So what we argue is that instead of dichotomizing clinical equipoise as either you have it or you don't, to compare traditional metrics, power, speed, and bias, with a participant level metric of how well they're doing, how much risk they're spending in the trial by having something probabilistically beneficial withheld from them. So to do this, we have counterfactual scenarios where you do nothing, no vaccination, or where you roll the vaccine out as if you assumed it worked in the smartest way you could with the resources you have. And comparing between those two scenarios, you have the amount of risk that is avertible. And now this is integrating the uncertainty in whether the vaccine works at all. So this is saying maybe 50% of the time it doesn't even work. And now between those two counterfactual scenarios, you can imagine any proposed design, any number of the designs we've discussed today, and it sits somewhere in the middle between doing nothing and vaccinating people as if you're rolling out. And so a proposed design will avert some cases, but it won't avert every avertable case because of the features of the design that are intended to gather information. So in some sense, you're spending avertable cases on information. And so now we have this metric of risk spent that we can compare, so that's on the x-axis here, to statistical power, where each dot here, or each symbol is a different trial design, and I'm not going to explain them in detail, but I can talk about it on the poster. And the summary is that the ways you can modify risk spent are by having a faster trial, so you could have a bigger trial to get information more quickly, so you can have post-trial rollout and post-trial access. And I'll cut myself off there. The last two slides are to highlight some work I've been doing with the WHO R&D initiative to develop a decision support tool to help people navigate the trial decision process, to understand how the context of a specific scenario interacts with all the different decisions you make while designing a trial. So this is a web applet. It's already online in a pilot form. And the idea is that groups of stakeholders go through it and look at how each decision you make in trial design interacts with epidemiology, the infrastructure, the vaccine properties, the sociocultural context, in a way where they can then take notes. And then as my final slide, I just wanted to plug a workshop that I run with several colleagues where we try to integrate this sort of simulation modeling and study design in a very multidisciplinary setting, and would welcome having more ethicists represented there, because we're trying to get as much diversity as possible. And I think it's really exciting to be at this meeting for me for that reason. So thank you.
Lots to discuss at dinner on that talk. Okay, so our next presenter is Dr. Kyaw Kyaw, and he's going to talk about, I think comes from Myanmar, and he's going to talk about ethical issues of alternative clinical trial designs and methods in Myanmar.
Good evening, ladies and gentlemen. I'm Dr. Kyaw Kyaw from the Defense Services Medical Research Center, Myanmar. I'd like to discuss about the ethical issue of alternative clinical trial designs and methods in Myanmar. Yes? Okay. So we have done so many research in Myanmar for decades, especially the cross-sectional descriptive design, and there are only a few phase 3 clinical trials, not too much clinical trials done in our country. So we have a research ethics committee. There are 18 research ethics committees in our country at the university and research institute. 2 are internationally recognized. And in our review process, the difference between the other country is the PI has to appear in front of the research ethics committees for every protocol. The reason why doing alternative research design in the world is a high cost of therapy development, and in rare diseases and conditions, there is no choice. We have to do that design, especially in the disease of genetic origins, psychiatric conditions, and HIV vaccine, like discussed in the morning. The ethical issues of this research are the vulnerable group, like the children, as in the case of genetic disorders and the psychiatric patient. There can be the physical risks, especially in giving the 2 drugs or the placebo group, and there can be the social risks in the case of HIV vaccine trials. And the challenges of alternative designs in Myanmar is researchers not doing alternative design because they are not used to it. And even the statistician and our research ethics committee are not used to it, although they are very effective and less ethical issue. The requirements of our research ethics committees are to practice to ensure there is no coercion to vulnerable groups like the children and the psychiatric patients, and to protect the physical risks as in a crossover trial design, because 2 drugs are given alternatively and in a placebo group. And other requirements are the justice for the host country, like our country. at a macro level, is it fair to do that research in our country? And choosing a cluster in the cluster RCTs. And the scientific validity, there can be inflated type 1 error because we cannot fix the covariates if one arm is stopped. The applicability in Myanmar, it should be applied in our country. As in the cluster randomized trial in the morning, it's logistically easier, and the similar projects are already done in Myanmar. But we have to raise awareness regarding the ethical issues. And another applicability, why it should be used in our country is, as in the morning, stepwatch cluster randomized trial. There are only a few healthcare system research in our country, but we already made stepwatch healthcare implementation, not the research, implementation, because of the scarcity of financial and human resources. So implementation already done. And we have to keep in mind that our tradition is a gatekeeper. These are the community leader, maybe the de facto leader or the de jure leader at the community. They are important for the acceptance because they can protect the right and welfare of the community, and they can point out the local interests of that area or our country. But we have to be aware that these community leader, if they are giving the consent is like the community consent. Sometimes the consent can act like the coercion for the consent of the other people, and sometimes the people can be confused, like a therapeutic misconception, because implementation are also done through the leader. So in conclusion, researchers should practice alternative research design because they are very effective and they are not used to it. And our REC should be made ready for the review process to facilitate the alternative research designs in our country. Thank you very much.
Our next speaker is Dr. Veena Swami, and she comes from India to tell us about alternatives to clinical trial design for Ayurveda in chronic non-communicable diseases in non-emergency settings. So to start, you push this button, okay?
Good evening. So we have an alternative to clinical trial design, but do we really have, and how are we going to assess that, especially non-communicable diseases in non-emergency settings like in Ayurveda? Because Ayurveda is, according to me, a system and not just a drug. So when we are trying to look at the efficacy, then we can't just afford to look at the efficacy, the result from the drug, because we are then looking at what is the system that it shows. So considering a difference between a system and a drug, so it is difficult for us to look at the impact of Ayurveda by measuring the drug levels. Now this is from Mangalore. I come from Mangalore Yanepal University, where people practice different types of medicines. There are enough Ayurvedic specialists, herbalists, including bone setters. And many of them don't go to orthopedician, but they go to bone setters to have the bone healed, the fractured bones and dislocated bones. So Ayurveda is, since ancient times, it translates as, it's age-old science, and it continues to be pertinent. And there are two methods of examination, one of the patient, which is Rogi Pariksha, one of the disease, that is Roga Pariksha. So in both these contexts, a detailed study has to be made before a doctor decides on what kind of treatment is required. So the context is much more important than the name of the disease itself, because when you're looking at a context, you're looking into patient as a whole. What is his body build like? What are his tastes like? What does he like and dislike? So identifying dosha becomes one of the most important things in Ayurveda. And it is this dosha that gets displaced, or it's in disequilibrium in case of illnesses. The dosha provides a blueprint for an individual for health as well as fulfillment. And Ayurveda describes three doshas, Vata, Pitta, and Kapha, and the combination of this, which is important for a healthy human being. Whenever there is a disequilibrium, it gives rise to a disease. So trying to treat a disease is setting right the equilibrium and not just giving a chemical component for that. So generally, Vata is that the quality that reflects the element of space and also in atmosphere. Pitta is that reflecting the elements of fire and water. Kapha is that which reflects the elements of water and earth. Now, every personality, depending on the physique as well as the questions that are answered, is into 3 groups. You could have a Kapha personality, you could have a Vata personality, or a Pitta personality. When the disequilibrium takes place, considering the background of the illness already, and then how you respond to certain questions put to you, what happens to you in certain illnesses, a physician decides what should be given to you. In other words, if 2 people are having the same ailment, it is not necessary that they will get the same treatment, because that again is dependent on your body dosha, which is Vata, Pitta, or Kapha. So, if a person is being treated for an ailment, now these are the detailed steps of examination, which almost are similar in allopathic medicines as well. That is Jiva, that's the tongue examination, the shape of the body, that's Akriti, the noise, how one responds to noise in illness, and the reasons with the eye, the sparsha, the skin, whether it feels warm or cold, the urine examination, the stool, and the pulse. Now these are examined in detail. And for each of this, it's like an algorithm or different sign symptoms that could be treated with a particular set of combinations of medicines, herbs, as well as massage and oil therapy. This is a simple, this is from the textbook of a urine examination. Now you can see that in case of Vata, certain qualities are seen. In case of Pitta, because one has to examine this in detail. It's not only microscopic, but more even the naked eye examination. Then a physician gets a better hang of the disequilibrium the patient is facing. So the treatment modalities here are non-specific modalities, oil massage, dietary alterations, and also some herbal combinations, and few specific herbal remedies. So when we look at a cure, it is not just one pharmacological agent here. And when we talk of non-specific, they're in expectation of a cure. Every patient responds in a unique way to the same dose and the medicine. In contrast to modern medicine, at average body mold, you get a similar treatment, a similar dosage. Whereas this, where the individuality and not the commonality is a measurable endpoint. But then, like in case of efficacy paradox, even if it's a clinical trial, in efficacy in modern efficiency is measured against a placebo. But this is an assumption that might be fallacious. In Ayurveda, non-specific modalities aimed at assisting the body to relocate its balance. And whereas the new kind of RPC is randomized, maybe a comparison trial, where a new scheme where outcomes from different studies should be merged together to assess a larger picture. In the existing hierarchical structure of research, error in the early levels will multiply, as in case of RCTs. Whereas if you have a circular structure, here what I mean in a circular structure is evidence coming out of efficacy, comparative effectiveness, and also patient safety. This depends on the documentation of the physician, Ayurvedic physician. So you don't take into consideration who you are examining at one point of time as a RCT, but how the patient has felt good throughout, and also the long-term prognosis. Alternate systems are well entrenched and accepted by public, but not by healthcare professionals. The need of the hour is to relocate these systems within the realm of science. The research model needs out-of-box thinking. And when I say this, I am also clear that there are challenges posed by Ayurveda itself. The preparations, the purity, the herbs, the adulterations, so that is a different set altogether. I'm aware that these are the challenges that one is facing, but yet one cannot just rule out Ayurvedic treatment because they are not in our set of evidences, they don't stand the test of time. So in the end, I would like to say that Ayurveda treatment, which has been for ages together, needs a better way of looking into and better way of understanding the sensitivity and specificity from the clinical trial methods. Thank you very much.
Our next presenter, Dr. Hany AlSaeed, comes from Egypt, and his talk is Paving the Way, Better Understanding of the Egyptian Research Ethics Committees. and Regulatory Authority for Alternative Clinical Trial Designs.
Thank you. Today I will talk about the ethics of alternative clinical trial design from Egyptian perspective. Egypt is a 29 million population with GDP per capita about $4,000 per year. This is very small. But the total number of registered clinical trial in clinicaltrial.gov, it's about 1,892. This is make it the second in Africa after South Africa. IRB in Egypt is about 56 institutional review board registered Office of Human Protection in USA. 42 of them are registered in NREC. NREC is Egyptian Network of Research Ethical Committee established in 2008 to establish harmonization and building capacity for IRB. It's operating under NGO called Society of Healthcare Development. We held biannual meeting. We have the 18 meetings last October. And we, from our point of view, that we need to have a knowledge regarding alternative clinical trial. We have also a national committee that inside the Ministry of Health, composed of 24 members that having meeting every 2 weeks. Every clinical trial international clinical trial must go through this IRB. The Egyptian system for reviewing of the clinical trial depend about institutional scientific committee for 30 days, research ethical committee institutional for 30 days, and scientific committee for the Ministry of Health called the National Organization for Drug Control and Research for 6 days, and the National Committee for 6 days. It makes about 6 months for reviewing process in Egypt is very slow. The possibility of alternative clinical trial in Egypt is very wide because we have unique study population and we need for individual tailored therapies in Egypt, and we have public health urgency. But until now, alternative clinical trial is not common. We had only one alternative clinical trial in Egypt passed from the National Committee. We must have training the IRB and the regulatory authority about alternative clinical trial design to help them to understand and review this type of research. So we decided to have a series of 45 minutes presentation followed by 1 hour focus group discussion. We began that in the last NRIC meeting. Point covered in the presentation about operational concern, recruitment and inducement, safety concern, fair inclusion and exclusion of the participants, safety monitoring, informed consent process on alternative clinical trial. Also include vulnerable population, the ethical acceptability of non-treatment control arm, and defining who is a research participant, and rule of gatekeeper, and risk and benefit assessment, and how to protect the vulnerable groups. Discussion will focus about the ethical dilemma of alternative clinical trial design and the sustainability of Egyptian culture and regulation. And this program will be supported by NREC. We began the last NREC meeting, like we said, to target 2 components of the Egyptian regulatory authority after that. In the last meeting, we had attendance of about 13 members of NREC. This is the preliminary result of this meeting. We ask about, do you hear about alternative clinical design? Majority say no. Can we control therapeutic misconception? Majority say yes. Can we have high qualitative informed consent? Majority say yes. Is waiver informed consent acceptable? Say yes. Can we consider group of the community as a research participant? Majority yes. Is the ethical intentionally infect healthy volunteers as a part of trial design? Majority say no. Can well-informed participant consent to any amount of risk if the social value of the study high enough? Majority say no. If alternative clinical trial design, we still lack about knowledge of alternative clinical trial. We need that. We need to help IRB and regulatory authority to understand the alternative clinical design, because I think it will be very beneficial for the Egyptian people. Thank you very much.
And our last presentation will be given by Gabriel Dao, and he will discuss the ethics of alternative clinical trial methods in an LMIC research, the Gambia Hepatitis Intervention Study Experience. This is the button to start. This slide's rolling.
Thank you. All right. Thank you very much. I am Gabriel. I'm a clinical research fellow at the MRC unit in the Gambia. I worked on the GHIS project, which is a collaborative study between the MRC unit in the Gambia, the WHO through the International Agency for Research on Cancer, and the government of the Gambia. Disclosure, when the study started in July 1986, I was but a little grumpy kid who had no role whatsoever in designing this study. The study was conceived on a background of a heavy burden of liver cancer in sub-Saharan Africa, with most of these cancers attributable to chronic infection with hepatitis B. Most of these Hep B infections were acquired in early childhood. At the time, there was this unique opportunity in the sense that there was a new hepatitis B vaccine that has been shown to prevent acute hepatitis B infections in certain groups of patients, and when given to infants, shortly after birth, it was found to be safe. So the study was set out primarily to find out if infant vaccination against hepatitis B can prevent liver cancer. There were other objectives of the study, of course, to see if it would prevent chronic hepatitis B infection and other forms of liver disease as well. We all know that liver cancer incidence usually picks up after the third or fourth decade of life, and therefore to properly assess this primary outcome, we would have to vaccinate children at birth and follow them up for a few decades. And this would mean that mass vaccination of kids with this vaccine, as far as preventing liver cancer is concerned, would have to be delayed by a few decades until we get the answer to this. Other limitations or other challenges at the time were that, like with most new interventions, availability of the vaccine was very limited and it was extremely expensive. One dose of the hepatitis B vaccine was more than all the other EPI vaccines available at the time. Therefore, the study designers designed a stepped wage study to quantify the net benefit of preventing liver cancer without having to delay the mass rollout of the hepatitis B vaccine. And the stepped wage also made it easier to implement the vaccination. The Gambia was a perfect place to do this study, given its relatively small land mass and population, and it had an excellent EPI vaccination program that covered the entire country and run by 17 different vaccination teams. And so the study would phase in the hepatitis B vaccine into different clusters attended to by respective vaccination teams. So at the first instant, one vaccination team would incorporate the hepatitis B vaccine into the EPI programs, with the other 16 serving as controls at the time. And at 3-month intervals, different groups would phase in their Hep B vaccination. And in the short term, after the end of the enrollment, we would assess whether it confers any protection against chronic hepatitis B within a few years, and 3 decades later, the incidence of liver cancer in the cases and controls. The ethical considerations at the time, like most step-wage designs, if eventually everyone gets the intervention, so it was socially acceptable, and it gave time to properly sort out the logistics, and by the time more kids would have to be on the vaccine, hopefully the cost would come down, making it more affordable. However, noting that the primary outcome would be measurable after 30 to 35 years, there was a risk, or there was a possibility that newer vaccines would be developed, and this current vaccine that we were testing would no longer be in use by the time we get the data, making it more or less useless. Other issues were that what would be the feasibility of tracing liver cancer cases and linking them back to the kids that were vaccinated or not vaccinated 3 decades previously, given the healthcare structure in developing countries. And finally, after 3 decades, after hopefully the vaccine being implemented in almost all countries in the world, what impact on policy would the findings have in the end, if it prevents liver cancer or if it doesn't, and so what? And so noting these issues, was it justifiable to delay the rollout of the vaccination by up to 4 years in some of the clusters that received the intervention lastly? Or would it have been better to do another study design where more kids get the vaccine earlier on? These are some of the issues that we discussed, and it's open for us to talk about more at dinner. Thank you.